Being able to image the exact same nanostructures on different instruments can allow scientists to determine their physical dimensions as well as chemical composition. If the samples are then processed, treated or modified, then the physical and chemical changes in the nanostructures can also be tracked. As you'll also see, examining the exact same nanostructures on different instruments can help scientists optimize imaging conditions, which can then help them generate better quality data. As you can see, the same 500 nanometer diameter carbon nanoring has been imaged using two different types of microscopy techniques: Scanning Electron Microscopy, or SEM, and Atomic Force Microscopy, or AFM. Both SEM and AFM produce magnified images of surfaces, but in very different ways. SEM magnifies images by firing electrons at the surface and detecting the electrons that are then emitted from the sample surface. AFM, however, magnifies images of surfaces by scanning a very small, sharp silicon tip across the sample, essentially tracing it. SEM can survey areas much larger and faster than AFM can, but produces flat two-dimensional images, while AFM has the advantage of producing images that are three-dimensional, as can be seen in the image on the right, where the carbon nanoring has been tilted slightly. This means AFM can measure the lateral dimensions as well as the height of nanostructures. So how is it possible to image structures so small on separate instruments? While it does require skill and patience, it is conceptually surprisingly simple. The nanostructures we are studying are carbon nanorings produced in the Vortex fluidic device, which was developed by Professor Colin Raston. Apart from the carbon nanorings, other materials such as small pieces of silicon and nanotubes are also present on the surface. We can use this material as markers to locate and relocate structures on the sample surface, as can be seen in the image where markers are indicated by arrows 1 and 2. By centering the SEM image on the nanostructure of interest and zooming out carefully, we can see additional material that can also be used as markers, as indicated by arrows 3 and 4. Once we reach a magnification that approximately matches the optical microscope attached to the AFM, we can then find our markers and move the AFM probe into position. We can then scan the sample surface with the AFM probe and locate our nanostructure. While we can locate and relocate the same carbon nanoring, we can also see that the AFM image shows what appears to be a nanotube in the bottom right corner of the AFM image. This nanotube seems to be missing from the SEM image on the left. This suggests that the imaging parameters used to acquire the SEM data may not be optimized. For example, the accelerating voltage used may be too high to produce the contrast necessary to resolve the nanotube, which can be easily seen in the AFM image. In this way, imaging the same nanostructures with different instruments can allow scientists to improve the way they generate data, which benefits their research and science in general. The instruments used to produce the data in this clip are part of Flinders Microscopy Microanalysis, which works in close partnership with the Flinders Institute for Nanoscale Science and Technology. I hope you find this video useful for your research.